In a second now. There we go. Okay, so the agenda for this week, we'll be discussing some dual payload ADCs platforms and why we believe this might be one very interesting modality for the next gen of ADCs. And we had some questions we were thinking to bring up and mention. Well, ask to you, Dan, mostly.
Yeah, that's fine.
Such as efflux pump proteins, upregulation of leach shock proteins, bystander retention, orthogonal pathways, block stress recovery.
Yeah.
A couple other topics which we had in mind. Do you want to start it off with something in particular, Dan?
Yeah, I think the best way to kind of look at all of those topics is
Ryan, sorry, but Ryan, if you want to jump in, feel free to jump in whenever you like. Also, if you have any thoughts, questions regarding this,
I'm happy to.
I'm mainly going to. Yeah, I'm mainly going to mainly going to talk about the deal making. But I'll jump in whenever I find it valuable.
Yeah. Okay, go ahead, Dan.
Yeah, I think I maybe made this point before when we talked about the ADCs before, but when you look at what's going on in the sector and all the topics you raised there, like efflux proteins, heat shock proteins, etc., it can sound quite overwhelming, especially when you start to think about antibodies that have multiple targets, multivalent, multi-payload, multi-linker, this, that, etc. So I always think the simplest way to try and think about the technology and also then think about what people need and why they're doing it is what problems are they trying to solve and where do the problems come from? And often that makes itself obvious when you take a step back and you think, right, what was the original technology? The original technology was an antibody that went to a cell and hoped to disrupt a cellular pathway from the outside. Those have achieved a lot of success, but then there's also a lot of limitation because they aren't actually providing necessarily a drug that gets into cells.
So then how does this differ from, say, a trispecific or bispecific antibody?
Well, in theory, it doesn't. In the technical element, a trispecific or a bispecific is using more points of contact on your cell. And when I say points of contact, I don't just mean gripping the same hook in multiple ways. That's like a multivalent. So a multivalent antibody, if you think about, I don't know, you're a rock climber and you're climbing a rock face, and you might be able to just grip on just like this on a little hook. But maybe if you're a multivalent antibody, you can hold on in lots of different ways on the same hold. If you're a bispecific antibody, you can hold and this hold, two different ones on the cell surface. If you're a multivalent bispecific antibody, you can hold both holds with multiple different, you're getting a better grip on both holds. Trispecific, 3 different holds. Multivalent. So
it's like you got better grip strength.
Yes, yeah. So grip strength is the multivalent aspect. And then how many holds you can grip is the bispecific, trispecific idea. And then that is all about targeting a certain cell population. So what you're looking for there is which cancer cells, is there a subpopulation of cancer cells that have X, Y, Z antigens expressed on the cell surface? Can we have an antibody that binds all of them? Can that antibody bind all of them at multiple sites so it really grips on tight? And the more levels of this you can add on, the greater specificity you get for that subpopulation of cancer cells, and therefore less off-target toxicity.
I was going to ask if you have, say, a trispecific or a bispecific ADC with a dual payload, would you might encounter some off-target specificity issues, perhaps?
Yeah, so I you can't ever rule out that that's not going to happen. Things bind.
And is that where the linker technology comes in?
The linker technology is really important for several things. So, do you know what? I'm going to come back to the linker if that's okay, because I think the framing for why the linker is important becomes a bit clearer when you think about what the actual molecules are doing. So, if you can direct an antibody to a cell population, you can then stick a drug on it and get the antibody to take that drug. And that is the antibody-drug conjugate idea. You're basically taking that payload into cells, it hits a target, topoisomerase, for example, or the
MMA.
tubulin protein, the DNA damage repair proteins, whatever it is, it goes in, it basically blows up a key pathway that the cell needs to survive. Okay, so why is that not good enough? Why do you need a dual payload on top of that? So the answer to that would be that the cancer cells eventually become resistant to the original drug. And that's because cancer cells are just rogue cells in a rogue population trying to do literally anything they can to stay alive. So if you're hammering them on one pathway, they will start to lean onto other pathways. Because it's not the individual cell that starts to change. It divides and it has a new mutation, and that new mutation gives it an ability to survive. It's more like a species evolving over time than a single cell making decisions. You probably killed that cell.
Yeah, like the coronavirus mutated.
Exactly. Cancer cells are very similar in that way that they can mutate rapidly. So if you're hitting an enzyme, it'll start to try and evolve away from that. So then you've got a dual payload idea, because you say, right, it's becoming, if we deliver this drug into cells through an ADC, it kills them for a while, but then they start to develop resistance. But if we also target that resistance mechanism, then we can shut down the pathway we were targeting and whatever was helping the cells become resistant. And that can take multiple steps. So that's when you were talking about the efflux proteins and the heat shock proteins. That is one route that's really popular, which is to say, how do the cancer cells try to get rid of the drug? So again, taking a step back, all drugs are poisons, and we have, as life forms, we have billions of years of evolving to get poisons out of our system. So we have efflux proteins that chuck small molecules out of cells. We have stress responses to try and help us repair damage caused by poisons. And what people who are in drug discovery are trying to do, it's like pinball in a way or something. You're trying to get this molecule into a certain type of cell and into where it can have an effect and kind of bypass all the security systems that have evolved for billions of years to get rid of toxins and poisons.
So it's like cat burglar almost.
Yeah, exactly. So, this is why a lot of chemotherapies were so blunt and brutal, because you are actually just saying, right, these are toxins and poisons, but this person's going to die if they don't take that toxin and poison, and we have to hope it kills the cancer cells faster than it kills the patient. Yeah. But now with selective targeting of cell populations, you can say, actually, we can make sure that poison only goes to those cells. But the MMAE and MMAF payloads that you see referred to a lot, they can't be given as a chemotherapy. They're too toxic on their own. They can only be employed as an antibody drug conjugate. So so these kind of new drugs can actually come out that, if delivered as a drug on its own, would kill somebody before you killed the tumor. But now you can actually target them. So this is where you start to think, why do you need to think about the dual payloads? You can start to say, right, what is actually causing the cancer cell to try and overcome this resistance? And one of them, you were talking about the efflux proteins. These are proteins that have evolved to, when toxins get into cells, efflux proteins take them out. And one of the techniques that's being brought in, I think it's Chengdu Labs, I think they're dual specific. They have the exatecan topoisomerase inhibitor, the traditional sort of blow up the DNA stabilization mechanisms. And at the same time, they have an RNA polymerase 2 inhibitor that is designed to jam up the cell's ability to make new proteins. So what would typically happen is the cell would detect a poison, and say, right, we need to start making more of the efflux proteins to get rid of that poison. But we're just with the other drug, you're throwing a wrench into that system that allows you to make more proteins to get rid of the poison. So that's the idea.
I do want to add something to that.
Mhm.
I've recently also read into that most of the dual payloads, they are sequential. So
Mhm.
you have one payload that's going to be destabilizing, and then the other one that's going to do the killing. But when it comes to the release timing, what's your thoughts on that, in terms of the effects on efficacy or safety?
Yeah, I think that's a really good question, and I think that is at the heart of a lot of this because with some of these combinations of drugs, it's not enough to just have the two go in and do the same at the same time. You need that sequential. This drug starts to have its effect, and then this one doesn't start to get released until it's needed. But also, it's a balance. You want it to come into effect when it's required, i.e., you want for example, RNA polymerase II inhibitor to come into effect when the RNA polymerase is going to start making these proteins. But on the other hand, drugs are metabolized in the body all the time. They've only got a certain half-life in the body. So you don't really want it to be wasted getting out of the cells and out of the body and metabolized when it actually is needed. So if you have this kind of sequential release, and this is where the linkers come in, Christopher, when you were asking earlier, because different linker types allow you to have that selection of when things are released and what order. So you could, for example, have an uncleavable linker for a drug that's going to block off the RNA polymerase II. It doesn't get released until the antibody's broken down and it's released with its linker. And by that time, your first drug had a cleavable linker, it's gone off, it's inhibited topoisomerase, it's had its impact on the cell, and just when it's starting to try and build resistance mechanisms, your other one's released and it goes in. This allows you to get the best of both worlds. But obviously, the immediate thing there is how do you actually balance the efficacy toxicity window, the therapeutic window, when you've got two drugs with potentially two different. And this is an open question. There isn't an answer to that really yet.
Yeah, like how does this impact the DAR and
Mhm.
CMC.
yeah,
Yeah.
CMC
That's what I'm
also a bit
thinking.
more tricky
Yeah.
to get it
It's quite complicated.
completely homogeneous, right?
Yeah.
For a homogeneous production, because you might get
Mhm.
4.5 and then 3.5 DAR
Yeah.
instead of you get a homogeneous 4.0 DAR. So that seems
Yeah.
a bit tricky. And also regarding to the linker, does it matter where you attach the linker and the payload? Does that matter?
I think, I mean, it must do. So you were saying with the DAR, I think an immediate problem is that as soon as you've got two drugs, even if you want to keep the drug antibody ratio low, you are twofold on any decision because you have two drugs to get in there. You have to then ask, do you want the same amount of each drug to get into cells? Do you want more of one, less of another? So that's going to maybe impact your ratio. But the other, I think I read today somewhere that only 2% of conjugated drugs actually make it into the cells they're targeting from a treatment with an antibody drug conjugate. So we're talking about a very small fraction. And when you have these linkers that could be cleaved in the blood, if 2% are getting to the target, but 0.2% is cleaving in the blood plasma, that can be an issue too. I think we talked about that before as well. So when you're dealing with the two payloads, from what I can understand, having a specific site that you are linking that payload to is much more important than it potentially was with the traditional ADCs, where you could have these kind of slightly less homogeneous populations where you had 3.5, 4.5 DAR across the population. As far as I understand it, you really need to know exactly how many molecules of each payload have been connected to your antibody, where they've been connected, and what the linker's like that's connecting them. Because a lot of these molecules are very hydrophobic. That just means you can imagine them being like sticky and they want to clump together. And that's when you were talking about off-target toxicity. When they become sticky and want to clump together, that just induces a kind of avalanche of sticking things together, like a snowball around in a hill. Things stick more and more and more, start sticking to proteins they're not meant to stick to. So all that effort you've put into having a multivalent, tri-specific antibody, but you've clumped up the antibody with hydrophobic molecules, and now it's sticking to anything anyway.
Yeah, it's like
So
someone throwing a wrench in the system and it doesn't
yes,
work. You might have the nicest strategy and well thought out plan,
yeah,
but if it doesn't go where it's supposed to, at the end of it, it doesn't matter.
yeah. So this is when you
Which is
see these hydrophilic linkers, and what they try to do is they have these long hydrophilic linkers that are effectively kind of like wrapping around and shielding the drug. It's kind of hard to visualize, but yeah, you can kind of imagine the
like
drug
a
tucked
rope.
away in close, yeah, and then wrapped around with a rope of the hydrophilic linker, and that kind of protects it. But then there's consequences to that, because when you've got a really long, complex molecule and you're trying to do some sort of chemistry to attach it to an antibody at a very specific site, the bigger and longer and more complicated it gets, the harder it is to actually carry that reaction out. So, everything's a balance. And there's, so there could be the best possible way to design it on paper, but when you're actually in the lab trying to make these kind of molecules, sometimes reality is just going to force you in certain directions, because this will happen, that won't happen, and so on.
So let's say someone has a huge like DAC payload they're gonna use for something like this.
Yeah.
How does that affect everything? How does it work if you combine it with a topo-1 payload? And
Yeah, I mean, I think degrader payloads combined with a traditional cytotoxic inhibitor payload. I'm not aware of anyone who's doing that yet. There may well be, and I've maybe not seen it.
might be some
But I
Chinese
think,
company.
yeah, probably. I think it's going to be tricky because they're already tricky molecules
Yeah.
because they're so big. So these hydrophobicity issues that we discussed, I mean, they tend to have a large linker between the two components of a degrader, but in effect, they're kind of a dual payload already. Not in the medical sense, but from a chemist's perspective, if the dual payload problem is that you have a molecule that's one inhibitor and a molecule that's another inhibitor, with a degrader, you've already got a molecule that binds to one enzyme and a molecule that binds to another, connected by a linker, and now you're linking that by a linker to another antibody with another molecule that's a cytotoxic inhibitor. At the chemical level, it's a triple payload. Not necessarily
like
medical, but at the chemical level, from the chemist's perspective, is synthesizing this. It's effectively, you could visualize it like a triple payload. So I think that's going to be tricky. Glues will probably be good. I imagine glues will be good in this space because they're small. They don't have those problems that the degrader would have. But that doesn't mean it's not possible, or someone won't do it. Someone I'm sure will manage it. But I would just imagine from a synthetic perspective, a glue as a single payload would be simpler with a cytotoxic inhibitor. But we're going to see it. People will find ways. There's, particularly with AI helping people to understand and run simulations that are far more complex than they ever could before, a lot of the difficulties might be able to be ironed out in advance. But yeah, it's such an interesting space. I think it's the next necessary evolution, because it's giving you that advantage to overcome resistance. And it's really cool from my perspective, because I think I was saying to you earlier, Christopher, my background is in chemical biology. I did a lot of what's called click chemistry, which is where you take chemistry that doesn't really exist in nature and you put it into proteins, and it allows you to do chemical reactions in places that you shouldn't be able to do that chemical reaction. And that is the underlying chemistry for a lot of these kinds of antibodies, where you can put in an amino acid that's not natural, that has a chemistry that you don't find in nature, and that allows you to do click reactions to have site-specific attachment of a linker with a payload, and so on, so on. And
Sounds
I
like
would
a lot
say,
of fun.
sorry?
Sounds like a lot of fun.
Yeah, when it works. When it doesn't work, it's not fun. That's why I know, like, I have the perspective that a lot of these things, when you draw them out on paper, they all work perfectly. When you actually run the reaction in the lab, you know, chemistry done on biology is uniquely difficult because the chemical reaction might work, but when you actually do it, everything on paper tells you it should run, and then the result just said no, it doesn't. And I had some bizarre experiences with molecules that should have behaved exactly the same, where one worked beautifully and the other one nothing. And there's no rhyme or reason, because biology just throws in unexpected hurdles that are very hard to pin down. You know.
Yeah, well, that's the complexity with biology versus just working with normal AI, working with tech. It's a bit more predictable.
Yeah, and I mean, I don't want to go off at a tangent, but it's one of my concerns with some AI applications in drug discovery is that ground zero is not there. You know, in the lab, you will sometimes hit a wall, and the wall is biology. And you can have things, you know, you can put a single mutation into an enzyme that should do this, and it just won't do it. And if life won't produce it for you, then it's very hard. So yeah, it'll be interesting to see. But you know, the AI tools will learn from these, and you know, they'll be trained on that data. If anybody who's watching this is interested in that kind of bioorthogonal chemistry, this kind of antibody drug conjugation, I really recommend reading someone called Caroline Bertozzi. She is like the biggest name in that field, and the work she's done is really, really amazing. And yeah, really recommend if you come across papers from her. Yeah.
Okay. Yeah. So, well, so like the conjugation strategy for all of this seems somewhat important. The linker included which payloads, where to bind them, which star. It seems like one can add a lot of complexity to all of this. Whereas before it was basically you had one payload or a few classes of payloads. You had the normal, like, standard antibodies you would typically use, and then see if it works or not, or how well it works. Whereas now it seems like we'll be able to experiment even more and find new, well, almost new classes of drugs, almost. Or new next generation ADC. Yeah, go ahead.
I do want to expand on that. So typically when we have conversations with licensees, a lot of the questions that we get is about docs and also CMC. I noticed personally that some of the companies that we represent are not really prepared for the CMC questions. So how, if any of you can answer that, how would you prepare for that?
Yeah,
but they would need to actually, like, if someone has previous experience with CMC, like they have been manufacturing, I would say that's a great starting point. If they don't have experience with trials or later preclinical stages, I would think it's somewhat difficult. Or what do you think, Dan?
Yeah, I would agree. I mean, it's not. It's at the cutting edge, right? So, you know,
most of it's preclinical. So
yeah, it's going to be tricky. I think at the CMC level, it's going to be tricky because there's a lot of complex chemistry that has to be done in parallel for not just the site-specific conjugation, but site-specific conjugation of two different molecules at two different sites. So you've got some chemistries that will run together, some that won't run together, but then you've got linkers that you want to be cleaved at different times. People will find a way. People, you know, we always find a way to do these things, and it's not beyond us. But I think that is going to be the trick. Is not that these things can't be done, but yield. You know, the chemists forever horror word. You know, what's the yield going to be? How much of these functional antibodies can you make in a CMC lab, you know, in the conditions that allow them to be, you know, drugs, you know, given to people? That's going to be tricky. That's going to be tricky.
Yeah, well, that's something that the pharma companies look at, because at the end of the day, it also has to make sense for them commercially, and not just okay, it's great if it works preclinically, it's great if it works in the clinic, but at the end of the day, they also want something that's profitable at the end of the
Yeah.
day. That's
Yeah.
preferable at the very least. I mean, if it doesn't make money, well, then it's pretty tough to get it to move forward, regardless.
I think you've hit the nail on the head. I think there's lots and lots of great ideas for what a drug could be, but if it's not something that can be manufactured in a way that it can make money and that can actually be delivered to the people who need it in a reasonable manner, it sometimes sounds cutthroat, but you do have to be aware that there's so many diseases and so many treatments needing made that you can't have everything all the time, and that you have to prioritize what makes sense. And it's very easy when people look at drug companies and say, oh, they make all this money on this drug that was successful, but they forget all the drugs that weren't successful and all the resources that go into all the failed drugs that didn't make it anywhere near the clinic. And those costs have to be covered, you know? So it's very easy to see the blockbusters and see the amazing success stories and think that's all that happens. But there's so much failure that has to be paid for.
There's a large graveyard if you think about it.
Yeah, absolutely.
Barely 10% actually reach phase 3 and pass phase 3. So I mean, it's most of the drugs do not make it all the way to approval for a reason. Just look at all the assets that are in the preclinical stages, and then you get to phase 1, you see a lot fewer than phase 2, 3. And these people don't see since they're not in the industry, they don't care too much. They just want free drugs, but sadly someone has to make these. And in order for everything to be functioning, someone has to make some amount of money from this. That's just how it is. Otherwise, there are no medicines. Because the governments won't pay for the development of these.
No, the governments can't. They can't pay for the development. You know, it requires a huge amount of investment and a huge amount of confidence, I suppose, and know-how. Like the tech. I mean, there's where I've worked previously, I know that there was prizes given to some preclinical drug centers for giving up on a drug early. You know, you get an award for demonstrating a level of awareness that this isn't going to go anywhere, so it doesn't make it into clinical trials and doesn't waste money and time and patients, you know, who are often quite desperate. And you're not putting things that aren't going to work into them. So, you know, they actually reward people and groups for knowing when to cut a loss, which I think that's a really important thing.
Yeah, of course. I agreed. And I was going to say something, but now I lost my train of thought. But maybe I'll get back to it later. But maybe Ryan, in the meantime, you want to touch on some deals?
Well, in the last 2 years, we've seen some deal making, specifically in the dual payload space, both M&A and licensing. One of the notable ones were, of course, by Lilly, Crossbridge Bio, with a 300 million acquisition. We also had Araris, which got acquired for its linker tech by Otsuka, which is actually basically an arm of Roche. And then we also had Innovent, which was out-licensing 12 programs to Pfizer for 650 million upfront, which to me sounds amazing, like a good deal for Pfizer, to be honest. And we've also seen some deals from Debiopharm, Samsung, and other multinationals. I did want to ask more about that, specifically, where does this specific interest come from? What's the biggest advantage here for a lot of these multinationals?
I mean, from my perspective, it's resistance to the drugs that they already have in their portfolio or competitors have in their portfolio. You know, if you already have, or your competitor has an ADC in a certain space, from a commercial point of view, if you can get the dual payload that operates in that space as well, then there's a population of patients that you're going to inherit when the original drug becomes resistant. Or they might never take that original drug if you can demonstrate matched efficacy or improved efficacy, well, improved efficacy with your treatment, So I suppose there's that looking into the future and seeing, okay, what has worked, but what wall is it hitting, and what payloads are actually meeting that wall and helping us get over it. And I think that's where they're looking.
I think that's also why we are seeing a lot of interest towards the platforms rather than specific programs. It's because they're looking beyond and they're looking at their own pipeline where they can improve or extend. So yeah, that makes a lot of sense. When it comes to payload classes, specific combinations of mechanisms, where do you see the most growing interest going?
Well, I mean, from my perspective, it's going to be wherever that resistance mechanism matches the payload that's being delivered. But there's a lot of different ways.
Depends on the goal.
A non-topo one payload. Mostly novel forms of payloads, and maybe they combine this with the MMA or a topo one. Something I believe would be of interest to a lot of parties.
Yeah. I mean, even subtle things as well that I hadn't. I mean, I read about this afternoon, and I hadn't really thought about this, but I'm sure there's a dual payload being developed. I can't remember where, but it's the MMAE and MMAF dual payload. Now, I had kind of viewed those drugs as synonymous, just analogues of one another, but actually they've got slightly different properties in their bystander effect and their ability to get in and out of cells. So actually treating those as a dual payload means that you're getting the same or slightly more efficacy from the target in the cell, but one of them stays in the cell longer, killing the cell it's in, and the other will drift out into neighboring cells. So there's a lot of subtle aspects like that as well that could make a pair of molecules a dual payload, partner.
So do you think that the conjugation experts will be of interest to the big pharma? They're gonna go out.
Yeah, absolutely. Because
already.
this is, I hate to say it because I really love chemical biology, but a lot of things that get designed in the chemical biology space never really make it into the real, even research application because they're so complex and so expensive that they reach proof of concept and then don't quite translate into an everyday tool. Some do, and that's great. So for me, I see this as a really interesting space for people working in bioorthogonal chemistry, or working in genetic code expansion and so on, because it's going to really need that expertise and it's really going to need that. I mean, I could go off on a real tangent here, but one of the complexities is when you're wanting to use these unnatural amino acids to have site-specific conjugation with different chemistries, you need whole enzymatic pathways installed in your antibody manufacturing system that puts that unnatural amino acid in, and you need one pathway for one and one pathway for another. So there's a lot of work being done on these kind of orthogonal pathways. I mean, that's a word that in this space, orthogonal is going to come up more and more and more. I don't know how comfortable people are with it outside of science. I know it gets used a lot, but effectively, when something's described as orthogonal, it really means that they don't come into contact with each other. So you can have a drug that has one target and a drug that has another, and they can kill the cancer cell in two synergistic ways, and they'd be described as orthogonal because they don't compete for the same target, and the resulting killing of the cell is separate from one another.
So
With these
someone who's riding a motorbike and a Formula One car or something like that.
Yeah, exactly.
Like they're in two different races.
Yes, absolutely, absolutely. And those can have an impact on one another, but they operate independently. So, when I've taught this as well, I have to, it's like orthogonality means this, and all these different contexts. But from what I'm describing there's very complex chemistry and biology knowledge needed to know how to put these unnatural amino acids in places where they're only in the one place and you have multiple different chemistries. That's going to take a lot of real expertise to achieve. So yeah, it's a very exciting time for chemical biologists, genetic code expansion enthusiasts, and I'm really excited to see where it goes, because it's nice to see some real medical application for some of these techniques that I think are really interesting. So if you ever want to talk about that, I'm happy to talk for hours on it. So
yeah, that's nice.
I do have maybe a silly question. It's something I saw somebody ask on X, and I just had to write it down. So why specifically go for a dual payload and not, let's say, go for 2 separate ADCs or an ADC with an oral? And I'm not sure if it's a silly question.
I don't think that's silly. I don't think that's silly at all. I think that's a really key question, because the idea would be, why not just deliver 2 ADCs to the cancer cell with 2 different payloads? And I think those things have been tried, like as combination therapies as well, or are being considered as combination therapies. I mean, or as a checkpoint inhibitor with an ADC. But from my understanding, the rationale behind not doing that is that if you've got a mixed population of 2 ADCs with the same cell targeting component of the antibody, so imagine you had the same multivalent trispecific antibody, but one with one drug and another with the other drug, they're going to compete on the cell surface, and they're going to outcompete each other. So you're going to get one bind or the other bind. Whereas when you have both payloads on the same antibody, it's going to bind and deliver them both. So I think that's the rationale there, because
that makes sense.
Yeah, when you've got like a tri-specific, it's great to say, oh yeah, it's got this hold, and this hold, but you're reducing the number of cells you can target with it, which is great, but you're also needing all 3 to be present in the same place, and the expression levels of the 3 will be different. And if you add that level of competition in as well, targeting that cell population would be very hard.
Okay. That's actually very makes sense.
Yeah,
right.
Yeah.
I saw nobody reply to it, so I was like, okay, that's
I think it's a great question. I think it's a really important question, because
this guy.
Yeah.
Okay.
No, I don't think that's a silly question at all. I think that's really important, because the logic there is like, why go to all these lengths to do all this complex conjugation and so on, when you could just treat someone with 2 drugs? And we see that
making a larger bullet or shooting the guy 2 times. That's what I was thinking.
Yeah. Exactly. Yeah. So yeah.
Nice. Well, yeah, I think that was. Do we have anything more to add before we wrap this up? I think I've felt like we touched on most parts we wanted to touch on.
I think so. Did. Yeah, I think we covered everything that I would have. I mean, there's always more, but I think we've hit the
most of the
key points. Yeah.
Okay. Thank you very much for listening. Thank you. Have a good one. Ciao.
Bye. Ciao.
Yeah.